What determines plants' extinction vulnerability? The impact of study scale. The future diversity of plant species will be negatively influenced by climate change. Scientists predict that more than 1 million species are on track for extinction in the coming decades. But what we can do to halt this crisis? Given that we wouldn't be able to protect all species, we need to focus our efforts on the species most susceptible to extinction. Understanding the correlates of extinction risk is therefore a very important step towards predicting the likelihood of extinction and developing effective conservation strategies. So, what attributes of species make them more vulnerable to extinction? It has been shown that plants' evolutionary history, phylogeny, their biological and ecological traits can help predict their fates. Phylogenetic relationships between species can play an important role in predicting extinction vulnerability. Certain families contain disproportionate number of rare species, suggesting heritable tendencies towards rarity. In general, rare plants are overrepresented in large plant families, while small plant families contain fewer rare taxa than expected. It has also been suggested that extinction risk is higher for young and fast-evolving plant lineages. Biological traits can also help predict plants' response to climate change, especially those related to reproductive success and environmental flexibility. Among life forms, a higher extinction risk has been suggested for trees compared to grasses and shrubs. Ploidy level can also contribute towards extinction vulnerability. Polyploidy, or whole genome duplication, is an important evolutionary trait that can enhance the environmental flexibility of species and their resiliency to climate change. Diploids are therefore more vulnerable to extinction than polyploids. Comparing mating systems, diastasis, possessing male and female flowers on different individuals, seems to be less adaptable to fluctuating or harsh environments and thereby increase the extinction vulnerability of species. Among pollination systems, biotic pollination by insects like bees can be more vulnerable to extinction than abiotic pollination by wind or water. Besides phylogeny and biological traits, the ecological traits represent significant predictors of the extinction risk. The ecological distribution of the species is often estimated by three indexes: abundancy, measured as the number of individuals found per study unit; range size, the area across which a species occurs; and habitat breadth, number of habitats or ecoregions occupied by the species. Species with a lower abundancy, species restricted to a smaller area, and species with a lower diversity of habitats are more likely to be at risk. There might be an interaction between biological and ecological traits. In other words, the impact of biological traits on the extinction vulnerability may depend on the ecological traits of a species, especially the habitat breadth. Considering the contribution of these factors and understanding their interactions will undoubtedly help to improve the accuracy of extinction predictions. However, an important aspect that is often neglected in extinction prediction is that the contribution of these factors and their significance might vary according to the scale at which extinction is studied. The conservation status of a species is often estimated at different scales: global, continental, national, local, or provincial, to direct policy and recovery actions at corresponding levels. As the scale changes, statistical relationships between these factors and extinction status may change and different patterns may emerge. Another aspect that can influence the accuracy of extinction prediction is the inclusiveness of the species to the study area. At each subglobal scale, two groups of species are distinguishable: species restricted to the study area and those not restricted to the study area. For species restricted to the study area, the conservation status often reflects the real status of a species. Species are either common or rare. However, this might not be the case for non-restricted species. A species that is common in the study area might be rare in the adjacent area or considering its entire range. And vice versa, a species that is rare in the study area might be common in the adjacent area or considering its entire global range. So, are the correlates of extinction risk of identical significance and accuracy at different scales and between regionally restricted and non-restricted species? To answer this question, we focused on the native flora of the Canadian province of Alberta and studies the correlates of extinction risk at both provincial and national scales and among provincially restricted and non-restricted species. The study was conducted within a phylogenetic comparative framework to account for the strength of the phylogenetic signal in extinction risk. We considered life form and ploidy as potential biological determinants of the extinction risk and habitat breadth as an important ecological trait. In general, no or little phylogenetic signal was found while studying extinction risk at national and provincial scales. However, after distinguishing between provincially restricted and non-restricted species, a higher phylogenetic signal was found in provincially restricted species, suggesting that the extinction risk is concentrated in certain groups. While life form didn't contribute towards extinction risk at the national scale, a lower extinction risk was found for shrubs compared to other life forms at the provincial scale and among provincially non-restricted species. We found a significant association between polyploidy and the lower extinction risk at the national level, but such association was not significant at the provincial scale. However, after distinguishing between provincially restricted and non-restricted species, polyploids showed a significantly lower extinction vulnerability compared to diploids among provincially restricted species. The habitat breadth significantly contributed towards extinction risk at both national and provincial scales and among provincially non-restricted species. Species with a wider habitat breadth were less likely to be at risk. We also found a highly significant negative interaction between ploidy and habitat breadth at both study scales. However, after distinguishing between provincially restricted and non-restricted species, such interaction was not significant. The negative interaction means that the impact of ploidy on extinction risk depends on the habitat breadth. Among species with lower habitat breadth, polyploids were less vulnerable to extinction, while among species with a higher habitat breadth, polyploids were more vulnerable to extinction than diploids. Summarizing, our study showed that the contribution and significance of variables are often scale-dependent. The accuracy of extinction prediction also depends on the study scale. The national extinction risk was more accurately predicted than the provincial extinction risk. However, we were able to significantly improve the model accuracy by focusing on the regionally restricted species, where the conservation status reflects the rare status of the species. Our study, therefore, emphasized the importance of study scale and discriminating between regionally restricted and non-restricted species for improving the predictability of subglobal extinction patterns.